Welcome friends, myself Shoptaporni from BioLive. Hope all of you are doing very well. Today we are going to discuss the solutions of previous year's question from the chapter Cell and Cell Division. This is the part 2 of this video. If you want to watch the part 1 of Cell and Cell Division chapter, the link is given in the description box below and also in the I button above. So before starting, I would like to request you to please like, share and subscribe to my channel by pressing the bell button given below. If you really like my content, please don't forget to like my videos. It inspires me a lot. And if you have any doubts regarding biology, you may ask me questions in the comment box, or you may ask me questions in my Facebook page BioLive Zoophilic, Facebook group BioLive Cell, or in my Instagram profile Shoptaporni BioLive. So without further delay, let's begin. The first question is, myelin sheath is produced by Schwann cells and oligodendrocytes, astrocytes and Schwann cells, oligodendrocytes and osteoclast, or osteoclast and astrocytes. The correct answer is option A, Schwann cells and oligodendrocytes. In the peripheral nervous system, the action of the neuron is enveloped by Schwann cells, and that is known as the myelin sheath. And in case of central nervous system, oligodendrocytes wraps the axons, and that is known as myelin sheath for central nervous system. Schwann cells, one Schwann cell can only wrap around one axon, but one oligodendrocyte can wrap around many axons. So this is the basic difference. So from this question we have known a lot of information regarding myelin sheath, Schwann cells and oligodendrocytes. Now coming to the next question, anaphase promoting complex is a protein degradation machinery for proper mitosis of animal cells. If APC is defective in a human cell, which of the following is expected to occur? See, anaphase promoting complex. What does it do? It is a protein degradation machinery. It just cleaves the link between the two chromatids. Two chromatids, and it separates two chromatids of a chromosome during anaphase. So if APC is defective, what will occur? The separation of chromatids, or the separation of chromosomes during anaphase will not occur. So the correct answer would be option C. Chromosomes will not segregate. Next question, DNA fragments are positively charged, negatively charged, neutral, or either positively or negatively charged depending on the size. It is a very easy question to answer. DNA fragments are always negatively charged because it bears phosphate group. It bears phosphate group which is negatively charged. Now the fourth question, the association of histone H1 with a nucleosome indicates transcription is occurring, DNA replication is occurring, the DNA is condensed into a chromatin fiber, or the DNA double helix is exposed. See, nucleosome is formed of histone octamer, H2A, H2B, H3 and H4. Each of them are present in double dose, 2 in number. H1 or linker histone is not present in the nucleosome. It just secures the DNA which is wrapped around the nucleosome. So when H1 is present or associated, then it indicates that DNA is tightly wrapped around the nucleosomes. That means DNA that means the DNA is now in condensed form. So it is condensed into a chromatin fiber. It is not relaxed. When H1 or linker histone removes, then the DNA, the wrapped DNA around the histone core protein also removes. That means the DNA gets relaxed. So when H1 is present, the DNA is condensed into a chromatin fiber. Now coming to the next question, Which of the following cell organelles is responsible for extracting energy from carbohydrates to form ATP? We know that the powerhouse of our cell is mitochondria, where ATP is formed. So the correct answer would be option D, mitochondria. Now the next question, which of the following are not polymeric? Okay, polymeric means those molecules, those macromolecules are formed by association of their monomers. See, nucleic acid. Nucleic acid is a polymer. Okay, polymer. Protons are polymers of amino acids. Polysaccharides are polymers of our monosaccharides. But lipids are not polymers. They are esters of fatty acid and alcohols. In case of triglycerides, they are esters of fatty acids and glycerol. So lipid is not a polymer, it is an ester. Okay. Now coming to the next question, which of the following options give the correct sequence of events during mitosis? See, here it is depicting mitosis. So see, in option A we will find crossing over. So obviously option A is incorrect. In option B we are not finding any crossing over. In option C also crossing over present, so it is also incorrect. Now the correct answer could be option B and option D. See what are written there. Condensation, obviously before when the mitosis starts, the chromosome gets the chromatin fiber gets condensed into a chromosome. Then nuclear membrane disassembles. Then chromosomes are arranged at the equator. Then centromere gets divided and segregation occurs during anaphase, and during telophase two daughter cells are formed. So obviously the correct answer would be option B. In option D they are given that condensation occurs, then arrangement at equator, centromere division, segregation, telophase. It is also correct, but here the nuclear membrane disassembly is not given, so it is not as apt as option B. So that is why option B is the correct answer. Okay. Now coming to the next question, mitochondria and chloroplast are semi-autonomous organelles formed by division of pre-existing organelles, and they contain DNA but lack protein synthesizing machinery. Which option is correct? Both A and B are correct. B is true but A is false. A is true but B is false, or both A and B are false. See, obviously mitochondria and chloroplast are semi-autonomous organelles. Why? Because they have their own DNA and they can replicate their own DNA. They also have their protein producing machinery. Okay. They have their own ribosomes, like mitochondria have mitoribosomes through which it can produce some proteins of its need. Okay, and other proteins are formed from the nuclear genome. That is why they are semi-autonomous, not fully autonomous, because all the proteins they need cannot be formed by mitochondria or plastid or chloroplast itself. Some of those are formed by nuclear genomes. That is why they are semi-autonomous, not fully autonomous. But so the option A is correct. But what is given here? Formed by the division of pre-existing organelles, and they contain DNA but lack protein synthesizing machinery. It is not correct. They do not lack protein synthesizing machinery because they have their own ribosomes like mitoribosomes. So option B is incorrect. So the correct option would be option 3. A is true but B is false. Okay. Now coming to the 33rd question. In a chloroplast, the highest number of protons are found in, it is very easy, the highest number of protons are found in lumen of thylakoids. Okay. According to chemiosmotic theory, for the formation of ATP, proton gradient generation is very much needed. And how protons are formed in the lumen of thylakoids? There the dissociation of water molecule leaves H plus ions, which are our protons. So that is why the lumen of thylakoids have highest number of protons. Okay. Now the next question, spindle fibers attach on the telomere of the chromosome, kinetochore of the chromosome, centromere of the chromosome, and kinetosome of the chromosome. Here two options are very confusing, kinetochore of the chromosome or centromere of the chromosome. Okay, option B and C. See, kinetochore, centromere is a part of our chromosome. Okay, it is the constriction, the primary constriction of our chromosome. Okay, but kinetochore is the protein complex associated with that constriction, that part of the chromosome. Okay, and spindle fibers attach to the kinetochore, not to the centromere. Yes, it is attached to the nearby the centromeric region where the kinetochore is present, but it directly attached to the kinetochore, not to the centromere. So the correct answer would be option B. Okay. Now coming to the 35th question, microtubules are constituents of what are given? Cilia, flagella. Obviously they contain microtubules, but peroxisome does not contain microtubules. So option A is incorrect. Then spindle fibers, centrioles, cilia. Yes, option B is correct. All of them contains microtubules. Okay. Now the complex of ribosomes attached to a single strand of RNA is known as polysome. Option A is correct. It is a very common and very easy question to answer, and it has also came in 2015. Okay, it is a very much repeated question, so it is very important. Now the next question, one of the major components of the cell wall of most fungi is chitin. Next, in meiosis, crossing over is initiated at, we know in meiosis prophase 1 there are 5 substages. What are those? Those are like leptotin, zygotin, pachytin, diplotin and diakinesis. The crossing over gets initiated at pachytin. Okay, at pachytin. So the correct answer is option A. Now the next question, which one of the following cell organelles is enclosed by a single membrane? See, it is also very easy question. Mitochondria, chloroplast and nuclei all are enclosed by double membrane, but our lysosome is enclosed by single membrane. Now next question, it is very much of informative type question. What water soluble pigments found in plant cell vacuoles are anthocyanins? Okay. Now, next question. Which of the following is not a characteristic feature during mitosis in somatic cells? See, obviously spindle fiber forms, nucleolus gets disappeared, chromosome moves, but synapsis does not occur. Okay, synapsis only occurs for the purpose of crossing over in meiosis. So it is not a characteristic feature of mitosis. So the correct answer is option D. Okay. Now our last question. A cell at telophase stage is observed by a student in a plant brought from the field. He tells his teacher that this cell does not look like other cells at telophase stage. There is no formation of cell plate and thus the cell is containing more number of chromosomes as compared to other dividing cells. This would result in the main statement from this paragraph is the cell is containing more number of chromosomes, okay, as compared to other dividing cells at telophase state. Okay, so whenever there is more number of chromosomes than the normal cell, then it is polyploidy. Polyploidy means, see, in case of humans what occurs, we are diploid, like right, so we have two sets of each chromosome. Okay, one is inherited from our father, one is inherited from our mother. In case of plants, polyploidy is very much common. They may have haploid genome, they may have one set of chromosome, two set of chromosome, or more than two sets of chromosome. Though presence of more than two set of chromosome is known as polyploidy. Okay, so here it is written that the cell is containing more number of chromosomes and cell plate has not formed yet at telophase state. So it is the result of polyploidy. Okay, it is a condition of polyploidy. So with this we end our today's question answer discussion here. If you like the video, don't forget to like, share and subscribe to my channel. We will come up with the rest part of this chapter's previous year questions in my next video. For the time being, thanks for watching and happy learning.